material, we have to alter the organic material in a way that facilitates that process. So in our stomach, we're going to use hydrochloric acid to soften and liquefy food. So for example, we would not be able to digest DNA if we hadn't exposed it to acid first in that process. Right. So the upper part, the mouth, is for biting, chewing, processing food. The pharynx and the esophagus are just transport vessels that are going to move food from our mouth to our stomach. In our stomach, we're going to do chemical digestion, mechanical digestion, liquefy food, and begin the process of moving things from the center of the digestive system into our blood, so absorption of water and alcohol. And then underneath our stomach, we have a small intestine that's very, very long, and so it's coiled up. So one of the really cool things about the digestive system that we'll begin to see tomorrow in lab with just the structure of the organs themselves, and then next Tuesday when we look at how the digestive system is constructed at the cellular level, we're going to see that the digestive system has this amazing varied ways of increasing surface area. And increasing surface area that allow us to do the digestion and absorption that we want to. So our small intestine is heavily folded up and coiled up in between our large intestine. And we divide the small intestine into three main parts. The upper part, which is the duodenum, the center part, which is the jejunum, and then the lower part, which is the ileum. And then the ileum attaches to the large intestine. And our small intestine is going to do chemical digestion and absorption. So we will have completed mechanical digestion by the time food exits your stomach. Right? And then the large intestine takes a very circuitous route around your abdominal cavity. Starting at the bottom part on the right side of your body, we have an area where the small intestine enters superior to this last kind of fold here, which is called the cecum. And then what comes off the cecum is a little appendage that we call the appendix. And then the food material goes up this part of the small and large intestine, so we call that the ascending colon. And then the large intestine is going to come clear across your body and go to the left side of your body, so we call that the transverse colon. And then the large intestine is going to drop all the way back down your abdominal cavity, so we call that the descending colon. And then what it has to do is it has to go back midline. So to go back midline, it forms an S-shaped bend to get back midline, and that's our sigmoid colon. And then the large intestine terminates in the rectum, which is a storage area for feces. And then it has a second opening to our mouth, which is our anus, which is the way we get rid of undigested food material, bacteria, and other things. So what we're going to do tomorrow in labs, we're going to go through those structures in more detail and begin to think about the processes that are taking place in there over the next couple of labs. So when we think about the digestive system, there are basically 7 processes that basically occur. So ingestion is the process of taking food material in. So that involves our mouth, our teeth, and other things that allow us to bite material off to take it in. Okay. So one of the things that we can think about our digestive system is the digestive system is essentially a tube that runs through you from your mouth to your anus. And then that tube itself is going to have different patterns to it, and that's what we're going to work on in lab next week. So anytime you have a tube in your body that has a space in it where food material would exist, the inside of the tube is called the lumen. And so food material passes through the lumen. Okay? So the tube is designed so that as we process material, we have a high amount of blood vessels that are associated with the wall of the lumen. So we have all these blood vessels. So one thing we need to make sure we understand is the difference between the word secretion that we're going to see through the digestive system and the word absorption that we've already talked about. Okay? So the reference point is to the lumen. So if we're going to move things that from the wall of the digestive system into the lumen, that is secretion. So that movement to the lumen is secretion. So notice that in 2 we say secretion is released by cells of the walls of the GI tract. Accessory organs would include things like water, acid buffers, and enzymes that are going to help us digest food material. Okay? So the other word that we used in the last slide then was the word absorption. So anytime we use the word absorption, what we're doing is referring to moving something from the lumen and putting it into the blood vessels. So this is secretion, and this is absorption. And that's the way you always have to think of that. And then once we do that, the next unit, we're going to use it in the same way. Again, secretion and absorption. So one of the things that we're going to see that we have in the wall of our digestive system is smooth muscle. So we're going to have a type of muscle that's going to help us move things through the digestive system. So 3 is a reference to the repeated patterns of contraction of smooth muscle. So we're going to use smooth muscle to help mix material within the lumen. And then we're also going to use smooth muscle to move it through the digestive system. So starting once we've swallowed the food all the way to the end in the rectum, we use smooth muscle to move material through our digestive system. So that pattern is referred to as peristalsis. So peristalsis is a rhythmic contraction of smooth muscle that occurs within the digestive system itself. So I remember as a child, my grandparents had a farm, and my grandfather would get 100 chickens every Easter, and then before winter, we would kill them all and divide them between our family. So the kids get to do fun things that the parents don't like to do. So one of my older brothers and I's jobs was to get all the chickens for killing. And if you do that, when it's fresh, the intestines quiver. And so I always thought that was really cool when I was a kid, but I didn't know why. And it wasn't until I took a class that talked about peristalsis. So that told me, oh, that's why they quiver. Because the smooth muscle just rhythmically contracts for a while, even after the chicken's been killed. And so it was a classic peristalsis that I was seeing as a kid. All right. And then what we're going to do is we're going to take big chunks of food and break it down into little chunks of food. So that process is called mechanical digestion, where we're going to take chunks of food and eventually liquefy it. And that's all going to occur between our mouth and ultimately our stomach, where the process of liquefaction is complete. And then what we're going to do is we're going to use enzymes, which are specialized protein molecules that break the bonds between certain atoms to take the large molecules and break them down into small molecules. So the use of enzymes to take large molecules and break them down into small molecules is called chemical digestion. And chemical digestion requires in our body enzymes for that to occur. And so what we're going to see is mechanical digestion is going to be completed by the time the food material exits our stomach. And we're going to start chemical digestion in our mouth, continue it in our stomach, and then complete it in the small intestines, in large intestines. So the sixth one was absorption. So we've already talked about that. Absorption is going to be taking material from the lumen and transferring it to blood vessels that are associated with the wall of the digestive system so that we can transport it all around our body. And then what we're going to end up with is material that we cannot digest. So the most common one is cellulose. So cellulose is what constructs the cell walls of most plant material and fungi, things that we ingest. And we lack the enzyme to break cellulose down. So it becomes a product that we actually can't digest. So it ends up as a non-digestible material. And so the way we get rid of non-digestible material is a process called defecation, which is the elimination of that non-digestible material from our body. So those are the 7 basic processes we're going to kind of look at through the unit and talk about it in some detail. So if we start with the top, we have a mouth. And associated with our mouth, we have some specialized organs, associated glands that we're going to talk about. So the mouth itself is formed by our cheeks to the side. The top of our mouth, the front of our mouth has bone. So the part that's in front on top of our mouth is called the hard palate. And the hard palate's actually rough. So if you actually open your mouth, used a flashlight in the mirror and looked at it, your hard palate's not smooth, it's rough. And so we use our tongue to push food against our hard palate to manipulate it between our teeth back and forth as we're processing it. And then we use our tongue to roll the food in a ball that we're going to swallow. So that's why if you eat white bread and peanut butter, it gets stuck on the roof of your mouth, because your tongue is trying to manipulate that by using it against the hard palate of our mouth. So it's very important that the front part of the roof of your mouth has that bone and the hard palate, because that creates the surface on which we're going to try to roll our food in a ball called the bolus. The back of it lacks bone, so we call that the soft palate. So we have a hard palate anteriorly, a soft palate posteriorly. The soft palate's important because when we swallow, the soft palate's going to be pulled backwards and upwards, and it's going to close off our nasal cavity, so that when we try to swallow food, it just doesn't come back out our nose, and so that it can be pushed down, right? And then the tongue is on the bottom floor of the mouth itself. And then we have a space between our lips and our teeth, and that space between our lips and teeth is called a vestibule. So we're going to see the word vestibule a couple times this quarter. And a vestibule is a opening inside of another opening. So it's a space inside of an opening, which is technically what a vestibule is. So what we're going to do is divide the vestibule into the inferior vestibule associated with your lower teeth and the superior vestibule associated with your upper teeth. And then the oral cavity itself is actually inside of your teeth. So that's the space inside of your teeth. In your mouth is the oral cavity itself. So if we look at that, and what we're just talking about, we can see that the hard palate actually does have ridges on it, which we actually use to manipulate food with our tongue. The posterior aspect of the palate, which is soft, has an extension downward called the uvula. And it's actually the uvula that's going to be pulled backwards and help us actually close off our nasal passage. There's actually a couple of cultures in the world where when kids are young, they cut the uvula off as part of the traditional culture. So it's very odd. But I haven't heard that they have problems with swallowing. All right, so then we're going to have a vestibule here and a vestibule there. We're also going to have ridges of, midline ridges of connective tissue that help connect things. And so the word we're going to use for a midline ridge of connective tissue is a frenulum. And then what we're going to see is we're going to have a frenulum associated with our lower lip. We're going to have a frenulum associated with our upper lip. And then we're going to have a frenulum associated with our tongue. And they're all midline ridges of tissue, connecting types of tissue. So we have an inferior labial frenulum, a superior labial frenulum, and a lingual frenulum, all in our mouth. And they're all midline ridges. The lingual frenulum is interesting because it can vary in people. And if it comes too far forward on the tongue, it limits tongue movement, and then those children have trouble learning to speak because their tongue won't move effectively. So we actually do surgery to actually cut the lingual frenulum in babies whose lingual frenulum is actually too pronounced. And then again, the oral cavity itself is this space between the teeth. And what we're going to see is toward the back, it's going to terminate with a couple of arches. And so we have a platoglossal arch, which is the most anterior arch in the back of your mouth. And then posterior to that, we have a platopharyngeal arch. But they actually have it mislabeled on this picture. So the platopharyngeal arch is this arch we're seeing back here. Not this. This line should go to there. So the platoglossal arch would be this arch. And then the one that you can see back here is actually the platopharyngeal arch. And that's because this mass of lymphatic tissue that we call a tonsil, the Palatine tonsil, sits between these two arches. The way they have it labeled, it's not between the two arches. That's one thing, so they have it mislabeled. We're going to talk about those more in the lab. Okay. Tomorrow. Okay, so when we're thinking about the process of ingestion, we use teeth to bite material off and to process material, break it into chunks. So that process is called mastication. So the process of chewing is technically called mastication. And it is a process involved with mechanical digestion. To be able to take food material and create a ball out of it eventually, we have to moisten the food, and we have to use a binder that's going to help keep it in a ball of food. So we use saliva as the agent that's actually going to help moisten food, and the mucus in saliva acts as a binder so that we could bind the food into a ball. So if you took a couple of soda crackers and don't drink anything at all, just start chewing them up, then every once in a while, open your mouth and look in a mirror, you'll convert that dry powder to a paste, and you'll eventually roll that paste into a ball as you get ready to swallow it. So that's the process that we use our tongue in our mouth for. So what we need is we have glands that produce saliva that's going to help us lubricate, bind, and liquefy food. So our biggest salivary glands are located just under our skin on the anterior aspect of our lower ear. So you can palpate yourself right here, and there's a glandular mass that you can feel on each side right here. So those are the parotid salivary glands. And they actually have a duct that crosses the masseter muscle, and then just anterior to the masseter muscle, the duct passes to the lateral aspect of the buccal cavity, about right here. And so that duct's called Stenson duct. You could go home and get a flashlight in the mirror and a lemon, and if you bite the lemon and then open your mouth, the change in pH in your mouth is going to stimulate the parotid salivary gland to release bicarbonate, which is a buffer, and so you can actually see it dripping out of the Stenson's duct with a flashlight. If you do that, you can actually see it dripping out of the Stenson's duct. So our other salivary glands are inferior in our mouth. So the submandibular salivary gland is the glandular mass you can feel right here, just near the angle of your mandible. If you palpate right here, you can feel two masses right there. Those are the submandibular salivary glands. And they have a duct that actually goes to the midline of your lower mouth, which is called Wharton's duct. So this is Borton's duct or the submandibular duct that you can see here. And then in that last picture where we're looking at the mouth open, here's actually the two little openings on either side of the lingual frenulum of the submandibular salivary glands. So some people can use muscle in the bottom of the muscles in the floor of their mouth and create pressure and squirt saliva. It's called leaking. And they can squirt saliva at you. And you can see it coming out of those two openings. And then under our tongue on each side, on either side of the lingual frenulum, we actually have the deepest of our salivary glands, which are called our sublingual salivary glands. And they have a number of little ducts that open called renovus ducts. So again, if you look at this picture, you can see that there are a number of little ducts coming off of the sublingual salivary gland. Go back to the last picture, you can actually see the little openings here for those renovus ducts on each side. So the one reason why when you go to the dentist, there's always more plaque that wants to build up on your lower teeth, and in particular on the inside of your lower teeth, is you have two salivary glands that are constantly putting saliva in the floor of your mouth. You have bacteria and the fact that you breathe in and out that concentrates that, and that's how come the plaque actually builds up more on your bottom teeth than your front teeth, because of the salivary glands that are in the bottom of your mouth. So years ago, and we have a vaccine for it now, so kids don't have to go through it. You would get an infection called mumps, and mumps would the virus that causes mumps would target the parotid salivary glands, and they would swell like grapefruit on the sides of your face, and you get a really high fever. So there was a fairly significant mortality rate with mumps, which is why vaccines are important. For mumps is the mortality rate that actually occurred because of the real high fevers that you would actually get. And so that was the parotid salivary gland that was affected. And so one of the things that we covered at the last 241 was the nervous system. And one of the last things with the nervous system that we covered was the fact that we have this autonomic nervous system. And the autonomic nervous system controls most of our biological functions where we don't have to cognitively think about it. And what we did is we divided the autonomic nervous system into sympathetic and parasympathetic branches. So what we're going to see is most of the digestive system is actually controlled by the parasympathetic branch of the autonomic nervous system. So for example, salivation, the ability to produce saliva, is created by the parasympathetic branch of the autonomic nervous system. So remember, the sympathetic branch was the fight-to-flight response or E situations. So if you don't like to get up and talk in front of people, you have to, what happens? Your mouth is really dry because the sympathetic branch overrides the parasympathetic branch and you stop producing saliva. So your mouth is really, really dry from that process. Right. So when we look at the salivary glands, then they produce a product that contains water. So the water is going to help us dissolve and moisten food. So what we're actually going to do within the salivary glands is move water out of our blood and create a product by absorbing or secreting. So what our salivary glands do? They secrete, right? Because we're going to go from the blood toward the lumen. So they're going to use water as one of their primary secretions. And then we actually make a mucus, which is a glycolipid that lubricates and binds food into that ball that we're going to create with our tongue called a bolus. We're also going to produce a bicarbonate. And a bicarbonate is a very important buffer. So if we eat any acidic food, we're actually going to try to maintain the pH of our mouth at roughly 7 by using a buffer. And so within our body, what actually happens because of our respiratory system So if we kind of put it together, so when cells are making ATP, what organelle is critical inside cells for making ATP? Mitochondria. So as cells make ATP, what the mitochondria do is they take carbohydrates and break it down. So the end product of that is carbon dioxide. So from the cells, we get these carbon dioxide from mitochondria. Your blood is mostly water. So as the carbon dioxide and water mix, we have an enzyme that actually converts a weak acid, converts it to a weak acid, which is called carbonic acid. And then what happens is the carbonic acid dissociates to form hydrogen ions and this bicarbonate ion. So this is the bicarbonate ion. So what happens then, once we form the bicarbonate ion in our blood, is we have sodium as one of our major positive ions that are in the blood. So the bicarbonate then interacts with sodium to form this compound, which is sodium bicarbonate. And so this is our buffer that we actually make in our blood and circulate within our blood. So what the salivary glands do is just take the bicarbonate off of the sodium, and then we secrete this bicarbonate as a very important buffer. And so we're going to see bicarbonate as an important buffer several places in our digestive system. So that's why I was saying if you bite on a lemon or something very acidic, it's going to trigger the parotid glands to increase the production of bicarbonate to neutralize the acid in the mouth. Okay. So the other thing that salivary glands do is produce an enzyme. And the enzyme is amylase. So by convention, chemists got together and the chemists said, well, what we're going to do is we're going to name all enzymes in a way in which somebody can know that we're referring to an enzyme. So all recently named enzymes are going to end in ASE. So that's always the ending that tells you it's an enzyme. And then what they do is they take the first part of the enzyme and describe what the enzyme is going to work on. So amylase is the enzyme, and what amylase is going to break down is the sugar amylose. So this is a sugar with OSE. The enzyme that breaks it down is amylase with ASE. So it's kind of a cool process. So we're going to work on that more in the third lab, where we're actually going to do some chemical digestion. But anytime you see in this class a compound ending in ASE, that tells you it is an enzyme, which is a protein molecule that helps us digest. So the other thing that we have in our mouth are enzymes that help break down bacteria. So our saliva produces enzymes that also help control the bacteria that can grow in our mouth. And those bacteria are called lysosomes. That help break down bacteria. So there's two critical things about health in relationship to your mouth is we use a bicarbonate to buffer our mouth to maintain a particular pH, which prevents things from being able to grow in our mouth. So it's really critical that the pH of your mouth is slightly basic. So if the pH of your mouth becomes too acidic, you'll get fungal infections in your mouth. And what prevents us from getting fungal infections is the fact that we're maintaining the pH in our mouth. Plus, we have these enzymes. So one of the things that's interesting is you take really high potent antibiotics. They'll kill the normal bacteria that live in our mouth, and we'll get altered species living in our mouth that create sores and stuff. in the mouth. And that's always a concern with antibiotic treatments, particularly very severe antibiotics. Okay. So then our tongue is essentially a muscle mass that is controlled by skeletal muscle that allows us to move our tongue around, and then controlled by muscle within our tongue that allows us to change shape of our tongue. So some people have a gene mutation for the muscles in their tongue, and they can't actually roll their tongue. So there are people who cannot roll their tongue. The tongue just stays flat. And that's a gene mutation to being able to control. So what we say is extrinsic muscles are the muscles that move the tongue around. So those are the muscles we learned in 242, styloglossus, hyoglossus, genioglossus, the muscles that we learned. Those are external to the tongue. So we call them extrinsic muscles. And they help us manipulate our tongue around. The muscles in the tongue itself that help us change the shape of our tongue, like rolling our tongue, are called intrinsic muscles because they're within the tongue. So we have two sets of muscles that we actually can manipulate, extrinsic muscles and intrinsic muscles that we're going to review in lab. We already talked about the lingual frenulum, which is that connective tissue connection underneath, and we talked about the fact that children that are born with too pronounced lingual frenulum, or used to be called tongue-tied, have trouble learning to talk. Our tongue surface is not smooth, it's rough, because we have all these projections on it called papillae. And that's very important because we use our tongue to manipulate food. So if your tongue surface was very, very smooth, then all the food would just fall off on the floor of your mouth all the time. And it would be much harder to manipulate. So what we do is we put sensory apparatus in these papillae. And so the most common papillae we have are very thin upward projections like this that are called filiform. And filiform papillae contain temperature receptors, touch receptors, pressure receptors. So they allow us to know what's in contact with our tongue. Then we have other types of papillae that actually contain taste buds. We're going to go over those in lab even tomorrow. So papillae are important because they increase the surface area of our tongue, and they create a rough surface for our tongue. And then we have glands on our tongue that are on the dorsum of the tongue, which is the top of the tongue. And they produce an enzyme called lingual lipase. So if we understand the pattern, then ASE is an enzyme, and this is what it digests. So lip is short for lipids, yeah. So a lipase is an enzyme that digests lipids. So when we take fat, when we ingest fat, we actually begin to break down the fat in our mouth. So we begin to break down carbohydrates, and we begin to break down fat in our mouth. Lingual lipase is probably much more important in babies that are breastfeeding or are on liquid diets of milk than it is adults, because lingual lipase is an enzyme that really helps break down milk fats in babies. All right, so just kind of as a review of what we were just talking about. So we have these upward projections called papillae, and the filiform papillae contain temperature, touch, pressure sensations. Fungiform papillae, which look like little mushrooms on our tongue, contain taste buds. On the side of our tongue, we have some folds we call folate papillae that contain taste buds. And then circumvallate papillae are ring-like structures that form a V at the back of our tongue that contain taste buds. So we'll look at those in the lab tomorrow. There's actually a real human tongue over there that's in the blue container that you can actually see all those papillae on, which is a preserved human tongue. And we have some models of So when we're looking at our tongue, this surface is the dorsum of the tongue. This is the apex of the tongue. And then we have these folds on the side here, which are the folate papillae. These are the fungiform papillae. All the real fine ones all over the tongue are the papillae, these are the Filiform Papillae, and then here's the circumvallate papillae at the back. So we have teeth to mechanically break down food, to bite food off. So when we look at a tooth, we can divide the tooth into three regions: the crown, which is the part of the tooth that extends above the gum; the neck, which is the part of the tooth where the gum covers; and then the root, which is the part of the tooth that extends down into the alveolar process of the mandible or the maxilla, the bone. So when you go to the dentist and they use the little probe, they call out the numbers 1, 2, and 3, 4. You don't like 3s and 4s, you like all 1s and 2s. What they're doing is looking at how far the gum is moving down the root of down the neck of the tooth. And the more the gum recedes down the neck of the tooth, the greater the chance that you're going to get cavities that are going to compromise the tooth. Because the crown is covered by enamel, which is one of the hardest substances in our body. And it prevents bacteria from being able to digest your teeth. The enamel becomes thinner and thinner in the neck of the tooth, so that as your gum recedes down the neck of the tooth, and that is exposed to bacteria now, it greatly increases the chance that bacteria are going to eat your teeth because there's less enamel. So that's the key. And then there is no enamel on the root of the tooth. So the enamel is only on the crown, and then it tapers in the neck of the tooth. So enamel then is the surface material on the crown of the tooth. What makes up the inside of the teeth is a calcified connective tissue, which is like a really soft bone material, which is called dentin. So what happens when you get a cavity is you have a little crack in your enamel, and bacteria got into that crack, and as soon as the bacteria get into the dentin, they can easily digest dentin. And so they digest this hole in your tooth because they can digest the dentin really easy, which is this calcified connective tissue. So we use the enamel to protect the dentin, which is the internal material of the tooth. Now, what covers the surface of a root of a tooth is a connective tissue-like bone substance called cementum. And that allows the tooth to be held in place in the alveolar cavity by little ligaments called periodontal ligaments. And then teeth are hollow. So at the center of the crown of the tooth, there's a hollow place in the tooth, which is called the pulp cavity. And then within the pulp cavity, we actually have blood supply and nerves that continually nourishing the dentin of the tooth. So that's why when you get a cavity, it really hurts, because the cavity eventually can eat a hole clear into the pulp cavity. And then once the hole is in the pulp cavity, then any liquids or anything get into that hole, and they stimulate nerves endings which make your teeth hurt. So that's one of the reasons why if you have a cavity, one of the things you notice is that the tooth becomes really sensitive to changes in the temperature, cold and hot. All right, so if we look at that real quick, while we were just talking about, you can notice how the enamel is at the tip in the crown, and you notice how the enamel tapers in the neck of the tooth to where it disappears at the end of the neck of the tooth. So as the gum or the gingiva recedes down your tooth, it exposes this area to bacteria, which increases your chances of cavities. So that's why they always use the probe to call out the numbers. And then what exchanges the surface in the root is cementum. So this is cementum. And cementum allows periodontal ligaments to connect to it from the periosteum of the bone. And that's what keeps your teeth in place. So if you ever had to have a tooth pulled, you can actually hear the periodontal ligaments break or pop when they're pulling the tooth out. You can actually hear the little popping of the periodontal ligaments. So the inside of the tooth is all made of dentin. That's that soft bone-like material. And then at the end of each root, whether you have one root, 2 roots, or 3 roots, there's a little opening called the apical foramen that the nerve and blood pass through. So the tunnel that the blood and nerve pass through the root is called a root canal. And then the nerve and blood get into this hollow area called the pulp cavity, which has got a semi-gelatinous material that maintains the relative nerve and vasculature. So if you get a bacterial infection inside of this, then your tooth really hurts. So what they do is they drill a hole in your tooth. They take a little wire brush-like thing and destroy the nerve and the blood vessel. And then they fill this whole material with the same material they use for making, for filling cavities. And so that process is called a root canal. And they actually kill your tooth. The tooth is dead after a root canal because they remove the blood vessels and nerves. So it's like you get this real painful tooth. What do? Just destroy the nerve, and it goes hurting. Right? So what's fascinating is we're born without teeth. Anybody wonder why? Mom knows why. So babies don't bite nipples off. So you're born without teeth. And then you start getting teeth in, and starting at 6 months, and you get one pair of teeth per month, basically, as you go forward. And you'll end up with 20 teeth. With your front teeth, the 4 front teeth on top and bottom are incisors. So if we start midline, we have 2 incisors going sideways. The third tooth going from midline out right here is a canine. And then in babies, the teeth behind that, the 2 teeth behind that are molars. So we use incisors for biting, and we use molars for crushing and manipulating food. So what happens, and so you may wonder, why do we have baby teeth and get adult dentition? There's 2 things to think about. Your skull grew. So if you got your teeth in as a baby, you would have extra space after your skull grows to adult size, and you don't have enough teeth to fill in the space because you only have 20 teeth. And when you replace them with permanent teeth, or what we call adult dentition, we have 32 teeth. So we increase the number of teeth by 12 because your jaw grew bigger. The other thing is that before dentistry, toothpaste, toothbrushes, there's a good chance that by the time you were in your teenage years, your teeth were already rotting out. And so they're actually replacement teeth to teeth that at one time rotted out. So I always think about that. I'm glad I live in the era of dentists, because having all those bad teeth would not be very quite painful way of existing. But they're actually replacement teeth because the others were rotting out. And so we have 32 teeth that we begin to replace it starting with the age 6, going to age 12. And notice that we have incisors, we have canines, but we add a new form of teeth that we didn't have up here. So we have molars up here and no premolars, but now we have premolars and molars. So we actually add these teeth because our jaws get bigger. And then we only have 2 molars here. We get 2 molars in early, then we have a third set of molars that oftentimes we don't have room for, and we end up having them removed, which we call wisdom teeth. Yeah. The wisdom teeth were probably replacement teeth. Again, before dentistry, your teeth were rotting out, and that was your last tooth you were going to get to finish your life with. And now we protect our teeth, and we don't have room for them in our mouth because our other teeth aren't rotting out. Yeah. So as you're going from midline to posterior, then we have incisors front, and so we have 2 on each side. So the 2 center teeth are called central incisors, and the next 2 are 2 called lateral incisors. And then the third tooth out is going to be a canine. So that's true in baby dentition and adult dentition, where we have the two incisors and the canine. So if you have a dog or a cat, the canine teeth are the really, really sharp teeth. So ours are sharper, but they're not really long and sharp like a cat's, because we don't grab little critters and hold on to them with our teeth and not let them go. So we don't have to have really long and sharp incisors. All right. So that would be the canines. So notice that in two-palate dentition we have the first molar, second molar. What's absent are these two teeth on each side. And so we have a first premolar and a second premolar. So we have 4 on our bottom jaw, 4 on our top jaw. So that's 8 of our teeth difference, is just the premolars itself for the 12. And then we get the first molar and second molar in early. And then we have this third molar that comes in very late, between 8, 17, and 25, that we call our wisdom tooth. And there's our other 4 teeth that we didn't have as a baby. So we have 20 teeth here, 32 teeth here. 8 of the 8 teeth that are different are premolars, and the other 4 are our 4 wisdom teeth, 2 upper molars and 2 lower molars. So there's kind of a pattern to it as well. If you look at roots, the incisors and canines have 1 root. Premolars, the first premolar can vary, but most commonly has 1 root. The second premolar has 2 roots. Then as we move into molars, the first molar and second molar have 3 roots. So there's a pattern to it, which is a connection. So the molars are grinding teeth, so they have the greatest pressure against them. So the increased number of teeth keeps them in place. The increased number of roots keeps them in place. Okay. We'll look at those tomorrow. So our throat is technically referred to as a pharynx. And the pharynx can be divided into 3 regions. The nasopharynx, which is going to be superior to our palate, our hard palate and our soft palate. An oropharynx, which is directly behind your oral cavity. So if you open your mouth, shine a flashlight in it, look in a mirror, the space behind your mouth you see is your oropharynx. And then we have a laryngopharynx, which is posterior to your larynx, which is what we commonly call our voice box or Adam's apple. And so what we essentially have then, if you were looking at a sagittal section, is we have a palate with the uvula, and our nose would be here, connected to that. And so this is nasal. And then here's where our teeth would be. So this is our oral cavity. So what's directly behind the oral cavity is the oral pharynx. And then down here we have a structure that allows air to enter our windpipe or our trachea. So this is the larynx. And so the space posterior to that is the laryngopharynx. So from superior to inferior, it's nasopharynx, oropharynx, laryngopharynx. And typically, we only get food material in our oropharynx and laryngopharynx. And we use the uvula and the soft palate to prevent food from entering our nasopharynx. So typically, our nasopharynx is only respiratory in function. And the oropharynx and laryngopharynx are both respiratory and digestive. And then what separates our oral cavity from our pharynx, particularly our oral pharynx, are those 2 arches we just talked about. So the most anterior arch is platoglossal. So the way to remember that, gloss is a reference to your tongue. So the one closest to our tongue is the platoglossal arch. The one posterior, which would be closest to our oral pharynx, is the platopharyngeal arch. So pharynx in relationship to oral pharynx. And then what sits between these 2 arches is a set of tonsils called Palate tonsils. So here's a picture showing what we were just talking about. So here's our hard palate, here's our soft palate, here's our oral cavity. So the space behind our uvula and superior to our soft palate is the nasopharynx. Here's the top of our larynx with a very important cartilage that we have called the epiglottis. The epiglottis is what we bring downward to close off our airway when we're swallowing food. So we can use the tip of our epiglottis as a landmark, the bottom of our uvula as a landmark. So if you're between the bottom of the uvula and the top of the epiglottis, you're in the oral pharynx. If you're behind the uvula and superior to it, you're in the nasopharynx. If you're posterior to the epiglottis, which is where the opening of your airway would be, then you're in the laryngopharynx. So nasopharynx, oral pharynx, laryngopharynx is really three parts of our pharynx. So in biology and anatomy, we always have all these high-tech words for simple things. So what we commonly call swallowing is technically called deglutition. Is the high-tech name for swallowing. Okay. And so what we're going to do when we're chewing our food is we're going to use our tongue and manipulate the food against our hard palate and eventually roll the food in a ball that we call a bolus. Right. So once the food is created into a bowl, the sensory apparatus on our tongue sends a signal to our brain that it's ready to begin this process of deglutition. Right. So what we're going to do initially is we're going to pull our soft palate superiorly and posteriorly. So notice we're going to close the opening between our nasopharynx and our oropharynx by pulling our soft palate posterior and superior. So now the uvula is creating a seal so that our nasopharynx now is closed off. And we're going to elevate our tongue. So the muscle that elevates our tongue like this was the styloglossus muscle that protracts the tongue. Retracts the tongue. And that's going to move the bolus into our oropharynx. And that's a voluntary stage. And then the importance of the two arches. So I want to go back to this picture. So the importance of these arches is that the arches are associated with the uvula and the soft palate. So if I take my uvula and I pull it upward and posterior, these arches are actually going to collapse. So when you're not swallowing, there's a wide opening between the arches. As you begin to swallow food, because the uvula is pulled up and posterior, the arches collapse and the opening between is going to narrow. And if food rubs against the arch walls, it gags you. So it's a mechanism to prevent you from taking in too large a bolus of food that's going to stick in your laryngopharynx. And if it sticks in your laryngopharynx, then you can't breathe. And you'd be going. And then somebody has to know the Heimlich maneuver, trying to create enough bolus to make you bring that back out. So the most common reason people do that is because they've been drinking while they're eating, and that deadens the sensations in your mouth. And they're eating something like rare steak or something that's hard to chew. It's the most common food material that people actually do that. And lettuce, leaf lettuce, because it forms, you swallow it easy, but it forms a film that covers that opening. And it's hard to get it off. And too, it's really hard to do the Heimlich, that's all that stuff lettuce. All right. So the other thing we want to do is we want the food material to actually go down our esophagus, and we don't want it to go down our airway. And so what we have to do is we have muscles that bring the larynx upward, and we also bring our epiglottis downward. And as the larynx goes upward and the epiglottis goes downward, we create a seal that then allows the bolus to be pushed into the esophagus instead of the airway. So the reason why, if you're going to have surgery and you're going to be given an agent to put you to sleep, you cannot eat. So you have to can't eat for 12 hours, is because it deadens all of this stuff. And if you vomit, because once the stomach gets low in oxygen, it irritates the stomach. So if you're under and you vomit, you're going to aspirate all of that vomit. It's just going to go down your airway. They used to kill people. So that's why you have to be without food for a period of time. So your stomach's actually empty, because your stomach's going to get irritated because you get a decreased oxygen level in your blood while you're under anesthesia. All right, so the first part is voluntary, moving the bolus from your mouth into the pharynx. And again, if it rubs on the arches, it's going to gag you and keep you from bringing it down, because you don't want it to get caught in this area here, because then you can't breathe anymore. But once the food is in your oral pharynx, then it becomes involuntary. So you may have had candy or gum and you were doing something and swallowed it. Once it's back there, it's not going to come back there, because it becomes an involuntary act instead of a voluntary act. And then it's going to go. So there have been people that like swallow diamond rings and stuff. And you have to wait 24 to 48 hours to because they're not going out. Because they're going to go out the other opening and they won't go out that opening. So the voluntary, so once we enter the pharyngeal stage, where it enters the oral pharynx, then it becomes involuntary. So as we said, the soft palate's pulled upward, the two arches collapse and form more of a slit. So the opening between the arches is called fauces. So what you do is you narrow the fauces in the swallowing process. So to make sense out of that real quick, we went back to this picture. If you wanted to do it, you could draw an arrow from the arch to the center, and then this is the fauces from this to this opening. It's called a fauces. Okay, so the other thing we do besides bringing our epiglottis down, and you can actually do this, make a swallowing motion. You put your hand right on your larynx here, make a swallowing motion. You can feel it go upward. So you have muscles that actually pull it upward, and you bring the epiglottis down. By the way, there's always fun terms like epiglottis. So epi is a prefix that means above, okay? So if you were to look down your airway, if you're intubating someone and you were looking down their airway, you'd have vocal folds that narrow your airway. So these are your true vocal folds. The opening between your vocal folds, the space that allows air to go in and out, is your glottis. So technically, if you look at a larynx, the way it's designed, the vocal folds would be here. This is a cartilage above this opening, so it's called the epiglottis, because the opening is called the glottis between your vocal folds. So that's where the word epiglottis actually comes from. And so we're going to swing our epiglottis backward. Then we actually have a ring of smooth muscle. So we have, throughout our digestive system, we have rings of smooth muscle that go around the lumen, and they close off openings, and so we call them a sphincter. So we have this ring of smooth muscle right here, so that when you're normally breathing, the epiglottis is closed off, so the air goes into your airway instead of into your stomach. So what you want to do when you're swallowing is close the airway off by swinging the epiglottis backwards and downward and bringing the larynx up, but you want to relax this smooth muscle so the bolus can enter your esophagus. So it's called the upper esophageal sphincter that allows the food to to enter the esophagus. So if you're not swallowing, the upper esophageal sphincter is constricted. That prevents air from entering your stomach and going into your airway. So it's kind of a good process. Okay, so once the food exits the laryngeal pharynx into the esophagus, then that's the esophageal stage. And then what happens is we have two sets of muscles in the wall of our digestive system. So if we were looking at the esophagus like this, we have one band of smooth muscle that runs this direction. And we have one band of smooth muscle that runs this direction in a circle. So we call them the circular and longitudinal layers of smooth muscle. And so what actually happens is the circular smooth muscle layer constricts behind the bolus, so the bolus can't go upward, and the longitudinal muscle shortens your esophagus, so that you actually bring your food down by constricting the circular layer and longitudinal layer. And that's called peristalsis, the rhythmic contractions that we get, which will bring the bolus all the way into our stomach. Because our stomach contains acid, our esophagus lining is not designed to tolerate acid. Then we also have to have a sphincter at the bottom of our esophagus that prevents the stomach contents from getting pushed up into our esophagus. So that's called our lower esophageal sphincter. So one at the top, one at the bottom. So one thing you'd see in patients, particularly as you age, this lower esophageal sphincter doesn't work as well. And you get gastric reflux up into your esophagus, and we call that heartburn. Yeah. So we have drugs that help prevent that from happening. So what actually happens is the more stomach contents you get up into your esophagus, the more eroded the surface of the esophagus becomes. And it triggers an increase in mitosis to try to repair the esophagus all the time. And it increases the chances of esophageal cancer tremendously. So people with chronic heartburn that is not managed oftentimes end up with esophageal cancer, which is a fairly deadly cancer. It's all triggered by erosion of the lining of the esophagus. So heartburn is something you should never let go. All right. So when we get to our stomach's going to have compartments to it. So we need one part of our stomach to store food if we eat an abundance of food at one time. And then we have to have another part of our stomach that's going to liquefy food. Then we have to have a third part of our stomach that's actually going to help move that liquefied food toward the small intestine. So the upper part of our stomach, we've got this kind of balloon area to it right here. And that's called the fundic region, or some people call it the fundus. And the way to remember that is that Thanksgiving dinner is fun to eat, but to store all that food, you have to have a fundus. Okay? So when you eat that large mass of food. So some animals are really kind of cool, like wolves in North America, the big cats in Africa, and Asia, lions and tigers. They all kill animals to eat, but their young don't travel with them. So once the young have stopped nursing, they have to be able to carry food back to them. And so what they do is a big cat like a lion can literally eat about 20 pounds of meat. And then they carry it back to the babies and regurgitate it to the babies. And so they actually have a fairly large fundus in their stomach, which is where all that raw meat is carried back to the babies. So I always think if I'm in a bad mood, like, wow, I'm not a baby cat. So my mother would have to, yummy. All right. So where the esophagus attaches to the stomach is called the cardia. And that's because it's just below the diaphragm from where your heart is. So it's very close to your heart. And that's also where heartburn will begin. cardia. And then this portion of your stomach is called the body of the stomach. So the body of the stomach is where we're going to liquefy food. So what we're going to do is we're going to convert that bolus, which is semi-solid food, into a liquid. And then as the food becomes liquefied, it enters a funnel-shaped part of the stomach, which we call the pylorus. So the pylorus is technically this opening from the stomach to the small intestine. So what we do, because it looks like a funnel, so the stomach is U-shaped, so it looks funny. But if we were to straighten it out, it would do this. It would do this to where the stomach ends. So the top of the funnel is called the antrum, the pyloric antrum. So that's where the food material would enter this. The tapering part is the pyloric canal. So we have a pyloric antrum and a pyloric canal. So the top of our funnel is the antrum. The tapering part is the canal. Then it's going to terminate in the pylorus, which is the opening that the stomach is. The pylorus is the opening that food is going to exit the stomach into the small intestine. So this is a cadaver stomach from a cadaver. And so this would be the cardia, this would be the fundus, this would be the body. And you can see right here, there's kind of a thickening. That's the antrum, and then the pyloric canal to the pylorus, which is the opening here. And again, we have a thick band of smooth muscle right here that prevents food from emptying from your small intestine to the large intestine until you want it to. So this thickening of smooth muscle is called the pyloric sphincter. And it helps control the emptying of our stomach. So what's pretty amazing is that the stomach, because it's U-shaped, has curves. And so the outer curve is called the greater curvature of the stomach. And then the inner curve is called the lesser curvature of the stomach. And what we have is we have connective tissue that connects to those curvatures. So connective tissue associated with the stomach is called an omentum. And so what keeps the stomach suspended from your diaphragm and keeps your stomach in place is connective tissue that connects to the lesser curvature of the stomach. So it's called the lesser omentum. And then we have this big sheet of connective tissue that comes off the greater curvature of the stomach, and it completely covers our small intestine. And so this big sheet of connective tissue coming off the greater curvature of the stomach that is covering our intestine is called the greater omentum. So this is a cadaver picture. Here's the body of the stomach. We can't see the lesser curvature, but this is the greater curvature of the stomach. So all of this stuff that contains a lot of adipose tissue right here is the greater omentum. So when you first open somebody's abdominal cavity, you cannot actually see the intestines at all. You just see this sheet covering with all this adipose tissue in it called the greater omentum. That's the greater omentum we're seeing. Okay. So one of the things that we said was that we're going to see all these cool ways in which surface area is increased. So if you have a surface and you want to increase the surface area, you can create upward folds. Or you could create downward folds. Now the difference we'll see microscopically is that upward folds create a broken surface. Downward folds create a nice smooth surface. But they're both ways to increase surface area, whether we're folding upward or downward. The stomach has both. The stomach has upward folds that create a broken surface area. And then on those upward folds, we have downward folds that create a more smooth surface area. Okay. So if we go back to this picture of the stomach, if you look at the lining of the stomach, it looks really broken up. So we have these really big upward folds that increase the lining of our stomach. And so they're called rugae. Okay. So those are visually something you can see with your naked eye. So if you actually cut a stomach open, the stomach is not a smooth surface. It's got all these ridges on it. And the ridges are upward folds, and they're called rugae. So what we're going to do in lab on right. So what we're going to do is we're going to see this really big fold on the stomach, which is a rugae. And then when we look at it under a microscope, we're actually going to see all these really small folds going downward from that. So we've got big folds that are upward folds, and many small folds that we can't see with our naked eye. We can only see with a microscope. Right. So this is one of the small downward folds, and so a downward fold in the stomach is called a pit. Okay. And then one of the things that we refer to the stomach is gastric. So the contents of the stomach are gastric juice. So gastric is a reference to the stomach. Right? So it's called a gastric pit. And so it's going to be a downward fold. And then what we're going to see is that on the inside of the stomach lining on the rugae, and then dropping downward into the pits, we have a bunch of modified simple columnar cells that secrete mucus. So a simple columnar cell that secretes mucus is called a goblet cell. So what we're going to see at the surface and in the neck of these pits are goblet cells. And goblet cells are designed to secrete mucus. We have to secrete mucus to prevent ourselves from self-digesting our own stomach. So because we have digestive enzymes in our stomach, and because we have this really strong hydrochloric acid in our stomach, we have to continually maintain a layer of mucus on our stomach that prevents us from self-digesting. So what happens if you don't produce enough mucus is you begin to digest a hole in your stomach, and that's called a ulcer. Yeah. So one of the most common reasons people get ulcers is a bacteria that eats the mucus. And when it eats the mucus, you no longer can protect your stomach, and you begin to digest your own stuff. Pretty amazing. Yeah. So in the upper part then, we have all these goblet cells that secrete mucus and protect our stomach. As we go downward, we're going to find functional cells that begin to produce the other products that we're going to need to help digest food. So as we go down the pit, we're going to end up with what we call parietal cells. And parietal cells actually secrete hydrochloric acid. So because what happens because we're adding carbon dioxide to our blood from our mitochondria, we make carbonic acid. Carbonic acid dissociates to form hydrogen ions in this bicarbonate. And then the sodium connects with the bicarbonate to form sodium bicarbonate, our buffer. And now we have a source of hydrochloric acid. Because now what those cells in our stomach can do is take the hydrogen ions and combine them with chloride ions and form a strong acid called hydrochloric acid. Yeah. So parietal cells take hydrogen ions out of our blood that were being produced because we're carrying carbon dioxide in our blood, combine them with chloride and create hydrochloric acid. And it liquefies food. So if you took like a turkey bone and you took an acid, so an easy one you could do at home, like a chicken bone or a turkey bone, a non-cooked one, and vinegar, and you just put it in a jar with vinegar, and then over time change the vinegar every day or so, you'll eventually make that bone rubber. And that's what our stomach does, is it liquefies things. So it converts solids to liquids. So that's why we need hydrochloric acid. Plus, it makes a very hostile environment for most bacteria. So none of the food you ingest is bacterial-free, unless you can figure out how to use an autoclave to cook with and transfer it from the autoclave to your mouth without contaminating it. Then the food you ingest has bacteria in it. Fortunately, there's only a dozen or so bacteria that can tolerate the high acid of your stomach and can create significant problems for you. The rest of it is all killed by the acid of your stomach. So the real key to our hydrochloric acid is the fact that it dissolves food and it kills most microorganisms that would be associated with food. Now, below that, we have chief cells. And chief cells produce an enzyme called pepsinogen. So here's the catch. So all recently found enzymes in an ASE, but unfortunately, enzymes we've known about for centuries were named before the ASE thing. So they aren't named that. And biologists are really bad about agreeing with chemists to change names. So we will find that we have some things that we're going to have to learn. So if you look at this word, pepsinogen, then and notice the OG in ending. And then the first part of it is pepsin. Okay, so protein digesting enzymes are very problematic for your body because the structural components of cells are proteins, which means that protein enzymes will digest all your cells. So we have to make sure that we can control where these enzymes are active. So what we do is we make them in an inactive form. So anytime the word ends in this, it's the inactive form of the enzyme. Okay? So what we're going to see is pepsinogen, trypsinogen, chymotrypsinogen. We're going to see a bunch of enzymes ending in OGN. So what we do is we have something that will activate the enzyme. So once the enzyme is activated, we drop this in here. So we end up with the word pepsin, which is the active form of the enzyme. Okay? So it's kind of a cool process once you figure it out. So what activates pepsinogen is hydrochloric acid. So we can produce this digestive enzyme that would digest all these cells in an inactive form, but it has to travel up out of the pit into the lumen before it gets activated by the hydrochloric acid. And it prevents us from digesting the wall of our stomach. Isn't that amazing? So that's why in a clinical environment, particularly if you were going into emergency medicine in an emergency room, and you have a patient coming in with a gunshot wound that perforated the stomach, or a knife wound that perforated the stomach, now you have all those enzymes that are outside, and they begin to digest all your organs. So what you have to do is flood the abdominal cavity with gobs of water, drain it, flood it with gobs of water, drain it, trying to dilute the enzyme mass, because you'll self-digest. Yeah. So if you had a gunshot wound or something that perforated your bowel, and you never did anything about it, you'd probably only live 4 or 5 days. And you're going to die from it because you're going to self-digest yourself. So, I'm done. Hard to leave you on that thought.